Good afternoon, and welcome to NC State's 7th annual 3-minute thesis competition, better known as 3MT. This is our second online version of the competition, and we hope to be back in person next year. I'm Peter Harries, Dean of the Grad School at NC State. And before we get started, I'd like to give a shout out to Joe Aldinger and Darren White, who are making the Zoom and live stream possible. We're incredibly excited to have you with us at this year's virtual 3MT. And a warm welcome to our extended Wolfpack family and friends tuning in from NC State, off campus, across the country, and around the world. There are indeed silver linings to being in the virtual environment, as it allows us to broaden access to this event and to make it much more accessible to many people across the globe. At NC State, our graduate students are leading research efforts across campus that have substantial impacts on North Carolina, the nation, and the world. Graduate students are diverse, collaborative innovators who put the think and do into NC State's motto. And as I like to say, graduate students really think and do. As a land-grant university, NC State graduate students are not only dedicated towards conducting innovative and cutting-edge research, but sharing that passion for discovery and change with the community. Whether that community be family, friends, politicians, lobbyists, schools, or neighbors, and inviting them to be part of the research happening here. Today, we're here to see 10 of those outstanding graduate students present their research in 3 minutes or less via video. The 3MT program is a research communication competition developed initially at the University of Queensland in 2008. The goal of the competition is to challenge graduate students to present a compelling presentation of their thesis and dissertation research and its significance in just 3 minutes to an informed but not expert audience. This effort has now spread across the globe, and there are universities everywhere that are engaged in this. But the real aim of 3MT is to develop academic presentation and research communication skills that supports the development of research students' capacity to effectively explain their research in language appropriate for a non-specialist audience. In this year's virtual format, presenters face the additional challenge of communicating their research with a single take video. To all the competitors, both those taking part in the finals today and those who submitted entries in the initial round, let me be the first to congratulate you. A job exceptionally well done under the circumstances. Once again, we're thrilled to present our second virtual 3MT program here at NC State. Thank you all for tuning in to support these outstanding graduate students. Now, the first thing that I want to do today is to thank the judging panel. Today's first and place winners were selected through the efforts of this group. A big thank you, first of all, to Dr. Katie Aman. Katie is the head of immersive experiences at the North Carolina Museum of Natural Sciences in Raleigh, North Carolina, where she has been part of the staff since 1996. In addition to department-wide managerial responsibilities, she also oversees program areas that include large public science events, online science interviews, lectures and town hall series, science cafes, and on-the-floor visitor programs. From 2017 to 2021, Katie was a board member of the Science Communicators of North Carolina organization. Before arriving at the Museum of Natural Sciences, she was a staff member for 9 years at the Museum of Life and Science in Durham, North Carolina. Our second judge is Dr. Rebecca Davis. Rebecca Davis received her master's degree from the University of Virginia in cell signaling and her PhD at the University of South Florida in cell biology. Rebecca spent several years as a postdoctoral fellow in the NC State Genetics Department. During that time, she participated in many of the activities and professional development opportunities offered by the NC State Graduate School and Postdoc Association, including things like presenting at symposia, competing in various competitions, and training through our Accelerate to Industry program. Rebecca is now an Associate Laboratory Project Services Manager at Q2 Solutions, where she manages genomic clinical trial testing, which has a $200 million portfolio supporting studies among 3 top-tier clients. Because of the support she received from the NC State Graduate School, those contributed greatly to her career development journey, and she is happy for the opportunity to give back and to be one of our judges. Finally, we have Dr. Haritha Malati. Dr. Haritha Malati is an assistant professor in civil and environmental engineering and currently the director of first-year engineering at the University of Delaware. She received her PhD from NC State and works in the intersection of engineering education, sustainable infrastructure, and community engagement. And also importantly, she was the winner of our inaugural 3MT competition at NC State, so she comes with a lot of experience in this arena. Now let me switch to some of the background and rules of the competition. Our competitors are only allowed 3 minutes, as the 3MT name implies, and they can only use a one-take video with one static PowerPoint slide to convey their story to you. So you can't use moving pictures or animations, no flashing lights, no interpretive dances, no creative special effects, no poems, just the brilliance of their word reflecting their research. The competitors you're seeing here today have advanced through a preliminary round of competition where they were selected out of a total of 27 presentations that were initially submitted. These presentations were initially judged to select those 10 that are moving forward today. And what's up for grabs today? First place is $1,000 and the chance to compete in the online 3MT regional competition at the Council of Southern Graduate School meeting that will be held here in Raleigh in the spring. And second place is a $500 cash prize and also has the potential to allow folks to compete at some of the other events that exist both at NC State and potentially at other arenas. And there's one last award, the People's Choice. And this is left to you as an audience to decide. So each audience member will be allowed a vote for this People's Choice Award. And this winner will receive a $500 cash prize. And it will reflect the person that this audience thinks is the most effective. So at the end of the string of presentations that you're about to see, make sure to cast your vote through the link that will be provided on screen at that point. You will then have 5 minutes to vote for that People's Choice, and then we will announce the winners live today. Okay, now that we all know the rules, let's get started. I'll be introducing the speakers and an interesting factoid about each one of them. Let's get started. Our first presenter today is Jerica Knox, a PhD student in psychology and a board member of the North Carolina School Psychology Association. And outside of academia, she is apparently a karaoke queen. Her presentation today is the enhancement of trauma-informed practice and schools through culturally responsive pedagogy, and her advisors are Scott Stage and Elin Hope, both in the Department of Psychology. Take it away, Jerica.
Childhood trauma. It affects children's learning and their behavior, as in we're in a classroom setting and we see a child and they're doing bad things and having temper tantrums, but On the inside, they're actually suffering from trauma, and the behaviors that we see are a result of that. And so trauma-informed practices utilize this knowledge that trauma affects learning and behavior to create practices that are sensitive to children's needs, like building positive student-teacher relationships. And they can essentially ensure that all children can learn in the school environment, including those who've experienced trauma. So what could be wrong with that? Well, the practices are currently being practiced within this one-size-fits-all approach, as in building that positive student-teacher relationship for one student looks the same for another. And yet if we tell a joke to one student and they're ha 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 laughing, but then we go to another student, they may be offended based on their own cultural background and lived experiences. And so that tells us that we need to use trauma-informed practices within a culturally responsive framework. Now, cultural responsiveness really takes this approach of thinking about children's lived experiences and their background. So they can essentially ensure that those same trauma-informed practices that we talked about can be implemented in a way that meets the needs of individual children, okay? And so trauma-informed schools and trauma-informed school programs need a little bit of help with their professional development. And so I've created a culturally responsive training program to implement within this trauma-informed school program. So we talk about things like historical trauma, racial trauma, inequities in schools, and the need for cultural responsiveness, as well as how to be culturally responsive. And so it helps to build up teachers' efficacy toward being culturally responsive, so yay. But more work is needed to make sure that teachers are able to really get from and interpret the content that's within these workshops. As in, we have teachers who may feel some discomfort with the content. Some may feel guilty or shame, and some may be uncomfortable with talking about race, which is essential to cultural responsiveness. And so future research and future work really needs to think about how we can best prepare teachers to engage in these conversations, as well as what can we do on our part to make sure that it is a safe and supportive environment. Essentially, these programs need to be culturally responsive and trauma-informed themselves so that teachers can go back and do the same for their students. Doing so will help to increase that cultural responsiveness, increase their ability to provide trauma-informed practices that meet the needs of students, and subsequently help our students to feel safe and thrive in the school environment. Thank you.
Thank you, Jerica, for that presentation. Our second presenter is Jack Jennio, a master's student in comparative biomedical sciences. Zach is applying to medical school this year with the hope of ultimately pursuing both a both an MD and a master's of public health. His presentation today is a peptide cocktail, the novel treatment for osteoarthritis. And his advisor is Lauren Schnabel from the Department of Comparative Biomedical Sciences. Take it away, Zach.
Osteoarthritis is a degenerative disease in the joint associated with aging and movement that can lead to severe pain. Almost 10% of people over the age of 60 suffer from OA. Maybe someone you know lives with osteoarthritis. For me, it's my grandmother. In horses, OA is naturally occurring, contributing to lameness in about 60% of horses, making them a perfect model to study and research osteoarthritis. The disease progresses when cells in the joint go into this inflammatory phase. Essentially, they stop producing all of those really good things like collagen, hyaluronic acid, the stuff that's in your face mask, and start producing the bad pro-inflammatory molecules. Currently, there is no cure, and the standards for treatment simply manage the problem, not improve it. Researchers found that if you take the elixir of life that is our blood and spin it down, you can concentrate all of the platelets with their great growth factors and host peptides and inject it right back into the joint to help assist with the healing process. This is called platelet-rich plasma, or PRP. My research mentor and team patented something called BioPly, which is a more refined version of PRP. And when tested against PRP, other treatments, steroids, and controls, the BioPly was able to downregulate the bad pro-inflammatory stuff and upregulate the good things like collagen and hyaluronic acid. Here's the roadblock. BioPly is not super easy to make. We require horse blood donors and have to spend about an entire day making it, which isn't bad, but what if we could find a way to make it more accessible? That's where I come in. Under the Schnabel Equine Sports Medicine Laboratory, my thesis focuses specifically on the individual peptides within BioPly that we've identified and what combination starts to create that desired BioPly effect. By researching this, we have the opportunity to synthetically make a peptide cocktail with all the beneficial peptides that could be distributed over the shelf, making it much easier than BioPly itself. So far, we've isolated some peptides, and the data is incredibly promising, and the tests start to rival the BioPly treatment. This research has the power to help entire horse populations, not to mention the treatments have translational effects into human medicine. Then maybe, who knows, someone in your life one day could be treated for their osteoarthritis with one of our famous peptide cocktails.
Thank you, Zach. Our third presenter is Rachel Nye, a PhD student in chemical and biomolecular engineering. Rachel loves swimming. She was a college varsity athlete and now continues to coach kids while she's in grad school during the summer. Her talk is entitled Untangling the Spaghetti of Polymer Thin Films for Advanced Technology. And her advisor is Gregory Parsons in the Department of Chemical and Biomolecular Engineering. Take it away, Rachel.
Thin film technology is important in every aspect of modern life, particularly in electronic devices and even in pasta dishes, but more on that later. These very thin layers of material have enabled tremendous advancements in technology over the last several decades. For example, consider a thin film coating on a battery electrode that enables longer lifetime in next generation electric vehicles, or creating a microchip for life-saving medical equipment. And when I say thin film, I mean very thin, approximately 10,000 times thinner than just one strand of hair. The reason for such rapid improvement in these technologies is because we've understood more and more about how the films grow and how to control them in a specific application. This means, of course, that to continue advancing technology, we also need to deepen our understanding of thin film materials. In my PhD, I worked to understand, at a very fundamental level, polymer thin film materials. And I'd like to talk about this in terms of something easy to understand and that would make my Italian grandmother proud. You guessed it, pasta. When you're making a good pasta dish, you have to consider how each ingredient will interact to contribute towards the overall flavor and appearance of the dish. You can control the type of noodle, the seasonings in the sauce, and how long each ingredient simmers together. Similarly, in my research, I can make a recipe for a polymer thin film, where I seek to understand how new ingredients will affect the overall film. First, I introduced two new organic molecules. Think of this like a type of noodle. And from these experiments, I came to understand that film structure is critical in determining how fast a film will grow and what properties it will have. Now, speaking of properties, the next step was to introduce a new technique. This analyzes the mechanical resilience of the film without damaging the material. So you can compare this to a new tool to eat spaghetti that doesn't get the tomato sauce all over your clothes. These new materials and analysis that I developed during my PhD have provided important information to understand how polymers grow and how to use them in applications. For example, creating better batteries for a more sustainable future. More importantly, this research serves as a springboard towards overall improvements in understanding thin film materials, leading to enhancements in healthcare technology, transportation, and of course, better tasting pasta. Thank you very much for your time, and I hope you learned something about thin films.
Thank you, Rachel. Our fourth presenter is Katie Christensen, an MFA student in creative writing. Katie has been working on this project for over 30 years. And her presentation is entitled Ethics for Human Subjects, Impacts of Misattributed Parentage. And her advisor is Jill McCorkle from the Creative Writing Program. Take it away, Katie.
When I was 15 years old, I learned through a high school genetics lab that the man on my birth certificate, my dad who raised me, was not my biological father, and he could never, ever know. My project, Ethics for Human Subjects, tells the story of misattributed parentage in the moment and aftermath of discovery. Over 30 million people have taken a DNA test. 2.5% of children are adopted, and somewhere between 4 and 10% of fathers are raising children who they mistakenly think are theirs. While my project is a novel, it is also in essence a case study of fractured identities, of the spectrum that lies between the innate desire to know one's biological family and the sincere reluctance to make those connections. On a craft level, I explore how mixed genre literature, performativity, and perspective can effectively represent the trauma that occurs in these moments of discovery through the telling of the story of three main characters. Katie Madrone, a fictionalized version of myself, is a precocious, science-minded outsider, a late bloomer, who writes letters and theatrical performances to cope with anxiety. Through interviewing family members and exploring artifacts, I was able to fill in the blank pages of a world of family secrets. Sasha Vale is based on interviews with the sister who I found at age 30. A well-grounded elementary school teacher in Portland, Oregon, she has always known of her adoptive status. A hefty folder of primary documents from the adoption agency and her biological parents helped me to tell her story. Finally, Kimbo St. Pierre, an actor and lighting technician in Olympia, Washington, does not become aware of being adopted until they take a DNA test at age 41, which also reveals an ethnic identity that it was not what they expected, sparking a journey for deeply buried roots. Kimbo is a composite character based on stories shared with me through a DNA Surprises group. Katie, Sasha, and Kimbo each grapple with what biology means in terms of their identities. Their stories tackle the following research questions. What catalysts tend to spark searches or refusals of genetic connections? How does the age of discovery impact the experience? What happens to relationships between family members who learn of a loss of genetic connection? What is the mental and physical toll of keeping secrets? And finally, how much of our being is tied to biology and how much is dependent on environment? Learning your genetic background is not what you expected can be a damaging and revelatory event for all parties involved. This project aims to contribute to the healing of families who struggle with such situations. Thank you.
And thank you, Katie. Presentation number 5 is from Mostafa Lubna, a PhD student in fiber and polymer science. She loves traveling, and in the 5 years that she's been in the US, she has already visited 20 states, including Alaska this summer. Her presentation is entitled Tie Up the Tiny Materials, and her advisor is Philip Bradford from the Fiber and Polymer Science program. The floor is yours, Mostafa.
Can you guess what is common in our laundry detergent and the James Webb Space Telescope? Yes, the use of nanomaterials. Materials having dimension in nanometers, approximately 100,000 times smaller than the diameter of a human hair. For their extraordinary properties, nanomaterials have very diverse use in cosmetics to therapeutics, in sports equipment to smart electronic devices, in environmental cleanup, and many more. The hype is because a tiny amount can tremendously improve materials' performance. On the other hand, there is a huge concern for nanotoxicity in terms of direct exposure to human and the environment. Some nanomaterials are non-toxic, like the zinc in our sunscreen, but most engineered nanomaterials have a certain degree of risk. Because of their teeny tiny size, they can easily interact with living cells and cause mutation or unknown damages. So, incorporating or embedding the nanomaterial in a safer matrix material like polymer or ceramic is a common practice that prevent the release of the nanomaterial in user environment and stop the nanopollution. But as the incorporation process is happening in bulk, there is a problem. The bulk matrix materials largely reduce the nanofunctionality. Imagine wrapping spaghettis with a blanket. Not seems very effective. Researchers all around the world are trying to find a better solution. Our research group works on utilizing the super strength, ultra low density, and excellent electrical signal processing properties of carbon nanotube, also called the CNTs. My research focused on strategic incorporation of CNTs in a safer polymer matrix to make a nanohybrid structure. I have developed a new method that coat the CNTs with safer nanolayer of polymer, like coating the spaghettis with olive oil, not with a blanket. This process is fast and well controllable. The safer polymer nanocoating not only keep the CNTs fully functional, but also help prevent the release of CNTs in dynamic use conditions. I have successfully demonstrated the use of this safe nanohybrid in 3 promising real-life applications: as a sensors in wearable e-textile devices, as a reusable super absorbent for oil spill cleanup, and in aerospace protection application. Whether we aim to build a colony on Mars or wanted to win the fight against cancer on Earth, the use of nanomaterial system is inevitable, and my research will contribute in developing a proper nanocoating method that will allow us to get the potential benefit from nanomaterials at a fullest extent effectively and safely.
Thank you, Mostakima. Our sixth presenter is Mamveen Kaur Ahuja, an MS student in food science. She loves reading, fiction, and traveling. And today she's going to be presenting on plasma treated water, a novel sanitizer for food contact surfaces. And her advisor is Deepti Salvi, also from the Department of Food Science. The floor is yours, Mamveen.
Hello everyone. Today I'm going to be sharing with you all a very unique idea which can solve the problem faced by food processing industries and is costing them billions of dollars annually. So what is that problem? The problem is that the biofilms can grow inside the stainless steel equipments which are used to either process or treat the food. But what are biofilms? They are a group of microorganisms which can be pathogenic for human health consumption, and they can make our food unsafe. And if we eat that food, we can get really sick. So it is very vital for us to get rid of biofilms as soon as possible. They get enough moisture and nutrients from the food which is being treated inside those equipments. So currently in the food processing industries, chemical-based sanitizers are used to inactivate them. But there is one problem associated with using them, which is that those sanitizers can leave harmful chemical residues on the food which is inside those equipments. And those residues have shown to be carcinogenic for human health consumption. So what do we do then? I have a solution for you all, which is that instead of using these chemical-based sanitizers, we can use plasma-treated water, which is totally chemical-free. So what is plasma? Plasma is the fourth state of matter after solids, liquids, and gases. And when we supply enough energy to third state of matter, we get plasma. So plasma is composed of powerful atoms, molecules, and electrons. And when we treat the water with that plasma, all these powerful atoms, molecules, and electrons get transferred to that water, and that water starts behaving as a sanitizer. There is one more advantage to using this plasma treated water, which is that it is totally an eco-friendly technique because we were using compressed air as a working gas to generate plasma. suggesting that this plasma treated water can be used as an alternative disinfectant in the food processing industry to get rid of biofilms, because one good news, when we treated the biofilms grown on stainless steel surfaces using plasma treated water, it was completely able to inactivate them. Thank you.
Thank you, Ma'am Veen. Our next presenter is Savannah Smith, a PhD student in civil engineering. Growing up, Savannah had a sheep who had 3 3/4 of a leg. His name was Gimpy. Today, Savannah will be presenting on Poop to Power, and her advisor is Francis de los Reyes III from the Department of Civil Engineering. Take it away, Savannah.
My name is Savannah Smith and I work with poop. If you're anything like my family, then when you think about poop, you maybe get grossed out. You just want it to be out of sight and out of mind. You definitely want me to stop talking about it. But when I think about it, I see so many potential resources in what some people think of as a waste. I want to take this unappreciated material and turn it into a source of power. If you live in a developed community, then when you flush your toilet, the wastewater most likely ends up in a wastewater treatment plant. Here, the water gets cleaned until it's good enough to be released back into the environment. But from this treatment, a large amount of solid material is usually left over. These solids are really difficult to deal with. They're heavy and they have to be trucked away. Best case scenario, we can use some of them to fertilize specific crops. But only the cleanest solids get to be fertilizer. More frequently, they just end up in a landfill somewhere. Wasting our waste contributes to greenhouse gas emissions, and it wastes a nutrient-rich resource that we could be using somewhere else. Now, you've probably heard that cow farts release methane into the air, which contributes to more greenhouse gases and then an increase in climate change. But what if we took those farts, captured them, and burnt them to create energy? This burning converts methane to carbon dioxide. And since carbon dioxide is a less harmful greenhouse gas than methane, this results in a decrease in the climate change potential of the gas by 1/30. Well, that's what I do with the solids from wastewater. If you take the solids left over from wastewater treatment and you put them in a sealed container, then you add microbes, which in this field we like to call bugs, the bugs will eat the solids and they produce methane gas. We can take this gas, burn it, and create power. The more the bugs eat, the more power we can generate. I'm trying to maximize this power by controlling which bugs are in the container and which are actually eating the foods. I do this by looking at the characteristics of the bugs that are already in the container and ones that could potentially get into the container with the poop. From this information, we can adjust certain parameters, like the amount of food or the temperature, and encourage certain bugs to eat and grow. If done correctly, we can reduce the amount of solids that we throw away and create a reliable energy source out of something that we would otherwise be throwing away. By doing this, we not only reduce the amount of waste that goes into landfills, but we're actually creating a power source out of a material that's readily available in many areas and is often discarded. Methane is an energy-rich power source that can be used in a variety of applications. So this is a potential solution to global waste issues, greenhouse gas emissions, energy demands, and ultimately, climate change and the health of our environment. Let's turn some more poop into power.
Thank you, Savannah. Our 8th presentation is by Madiha Nazir, a PhD student in microbiology. She got married over Zoom, and today she'll be talking about microorganisms, the powerhouses of the future. And her advisor is Jose Bruno Barqueña in the Department of Microbiology. Madiha, please start your presentation.
Remember the recent fuel shortage in North Carolina and the panic that ensued? Incidents like this are a wake-up call that demands for the development of alternative sources of energy. For example, exploring microorganisms' natural ability to produce biofuels. My research contributes to the solution of this problem by introducing a bacterium from Clostridium species. It is nurtured in a special chamber filled with nitrogen and hydrogen gases and looks like small rods under a microscope. This microorganism has innate ability to eat sugars and convert them into industrial chemicals such as butanol. Butanol has ideal chemical and physical properties to be used as a transportation fuel and has many applications in textile, construction, and cosmetic industries. The demand for butanol is only going to increase in the days to come. According to an estimate, the global market size for butanol will reach $4 billion by the year 2025. My goal is to meet this rising demand by channeling the abilities of Clostridium. There is, however, a catch. Working with this microorganism is not easy. It needs expensive laboratory equipment to grow in an environment lacking oxygen, and the quantities of butanol produced are not sufficient for use on a large scale. In order to address these bottlenecks, I have developed a method to genetically modify this organism. The modification will allow us to grow this bacteria aerobically, simplifying the experimental conditions and saving a lot of costs. I've also identified prospective genetic targets that can be manipulated to improve this microbe's capacity to produce butanol. It gives us the opportunity to develop an industrially suitable version of this microorganism. My findings will also open the doors for future researchers to meet the ever-increasing energy demands. When it comes to the potential of microorganisms, there is still a lot to explore. Many known and unknown organisms possess unique traits for enabling the development of biotechnologies to produce fuels and commodity chemicals. Not only this, these special creatures can save us from the devastating impacts of climate change. Along with wind, solar, and tidal energy, microorganisms can help us reduce our carbon footprint. These little creatures, isolated from soil, quite literally be the powerhouses of the future.
Thank you, Madiha. Our penultimate presenter is Juan Rivera, a PhD student in food science. His research focuses on sweet basil plants, and they are commonly grown in hydroponic systems in the US. And related to that, today he'll be talking about cold plasma technologies for hydroponic sweet basil growth. His advisor is Deepti Salvi in the Department of Food Science. Wen, take it away.
Hello, everyone. My name is Juan. I'm a PhD student in food engineering. Today, I want to talk about our research, cold plasma technologies for hydroponic sweet basil growth, which can be produced on location and on demand. But what is plasma? Well, plasma is the fourth state of matter, and we have seen it in nature in the northern lights and in lightning. You see, when you have a gas, such as air, and you add enough energy to it, it can transition into a plasma phase, where it contains reactive nitrogen and oxygen species, such as nitrate. This component, nitrate, is relevant because of the commercial fertilizer industry, which uses it in their formulations to grow plants. However, in contrast to commercial fertilizers, plasma can be produced both at atmospheric pressure and at colder temperatures, such that if you have a plasma source and expose it to a liquid, we can transfer the nitrates to that liquid and possibly use it to grow plants. This is relevant because of the $9.5 billion hydroponic industry, which uses a liquid called nutrient solution to grow plants without soil, as you can see in the picture on the left. Since this liquid is recirculated throughout the whole system, if it becomes contaminated by microorganisms harmful to plant health or to human health, then the hydroponic farmer may lose their whole crop. And if they didn't catch the contamination on time, then their produce can be a vector for foodborne illness spread. An example of this is sweet basil, which as early as February of this year was involved in a recall due to the risk of foodborne illness to the consumer. So we knew we wanted to help hydroponic farmers, and we knew from the literature that plasma could inactivate microorganisms or could help with plant growth. But we saw a gap in terms of plant quality. So that's what we did. We look at the plant growth, the plant quality, and the sanitation of the growth environment of controlled sweet basil plants compared to sweet basil plants grown in plasma treated nutrient solution. Our results showed that we used a similar amount of resources in terms of land and energy and water. But looking at the plant quality and plant growth, our plants were more attractive because they were taller and they had more leaves. This is especially exciting because conventional sanitizers in the hydroponic industry can harm plant health, but in our case, we actually improved it. Furthermore, when we looked at the sanitation of the growth environment, we saw that plasma was better at activating microorganisms such as algae and non-pathogenic E. coli compared to the control. Therefore, we concluded that plasma is a promising alternative for hydroponic farmers to grow both safer and higher quality produce. Thank you for listening. And also, I would like to thank our collaborators at Drexel, Rutgers, and the USD and NIFA, without whom this study could not have happened.
Thank you, Wen. Our final presenter today is Shohan Shohan, a PhD student in industrial engineering. He loves long car rides and exploring new cities. And today he'll be talking on solving human organ development challenges. And his advisor is Rohan Shearwaker, also from the Department of Industrial Engineering. The floor is yours.
Every day, 17 people die in this country while waiting to get an organ transplant. Every 9 minutes, another person is being added to that waiting list. So how can we solve this crisis? Clearly, waiting for someone else to donate the organ isn't working. An effective alternative would be fabricating those organs. Using 3D printing and other revolutionary methods, we are on the verge of developing human organs in laboratories. Did you know all the organs in our body generated from a single cell type? These are called the stem cells. So if you want to develop an organ for a patient, extracting his stem cells and converting them to organ-specific cells is the way to go. Suppose the patient needs a new heart. You collected his stem cells, added them to the growing heart, and followed all the protocols to convert them to heart cells. Now, how do you know that the cells are alive deep inside that new heart and if they have converted? You cannot just slice the heart in half to check, right? Because then you don't have an intact heart anymore for the patient. That's where my research comes in. I am developing an artificial intelligence or AI-based approach to check the presence and conversion of stem cells. As long as the cells are alive within the heart, their presence will be captured by a sensor that measures the electrical properties from outside without disturbing the organ. Now, knowing that the cells are alive, the next question is, have they converted into heart cells? I observed in each stage of the stem cell conversion, the electrical properties change a bit. This results in change in signal pattern. So in our case, the patient stem cells would show one kind of pattern, while the converted heart cells would show a slightly different pattern. It's very difficult for humans to track those changes, but my proposed AI approach detects them with very high accuracy. Just imagine the possibilities it represents. We can control the organ growth precisely while developing multiple organs for multiple patients in parallel. I strongly believe my research finding will play a significant part in ending the organ shortage forever. Thank you.
And thank you, Shohan Nuzaman. Okay, at this point, we're ready to start the process of evaluating the People's Choice. And we've now heard from our 10 finalists in the 3MT competition. And it's time for you, our audience, to vote for the People's Choice Award. So please take out your phones or open a tab in your browser and use the Go link, go.ncsu.edu/3mt-vote and select the name of your number one competitor. Remember, while all the presentations were excellent, you can only vote for one of these, and your email address is required when casting your vote. The voting will be open for the next 5 minutes. As you can see, we're down to about 4.5 minutes already, and we will announce the People's Choice winner along with our first and second place winners once those votes have been tallied. While you're thinking about your People's Choice and voting, I do want to take a moment and thank some people without whom this competition would not have been possible. First of all, the graduate school professional development team, which plays a huge role in coordinating the 3MT process, ranging from judging preliminary submissions to coaching the finalists and arranging the judges. So a big thanks to Laura DeMars, Katie Homar, Joe Aldinger, Vanessa Dorio Anderson, and Chris Smith, all members of that PD team. Another special thanks goes out to Darren White for his help with the communication aspects as well as the logistics of today's contest and in promoting it through social media. Finally, we want to recognize the mentors and advisors who've helped our finalists get into today's contest. We've witnessed the evidence of your work and support today, and you should each be proud of these outstanding students, researchers, and ultimately competitors for today. So a hearty thanks to all of you mentors and advisors watching today, and those of you who played a role in the development of these outstanding grad students. Now we have about 2.5 minutes left. And so if you haven't voted for your favorite speaker yet, please do that. The time is running out. So we will take a brief break and reconvene once we know the winners. See you soon. Okay, well, welcome back, everyone. And we're going to do one important thing before we announce the winners. Let's give all of these competitors a round of virtual applause. I mean, it's really impressive, the work that they've done. And it's just great to see the research that's going on here and also their ability to translate their research into a way that's very understandable, even for people outside the discipline. So just wow in terms of what they've been able to accomplish. Now, at this point, I want to announce the People's Choice winner. And I want to thank the audience again for voting for your favorite speaker. And our People's Choice winner this year is Zach Jennio. Congratulations to you, Zach. A job extremely well done. Now, getting to our second place winner and our first place winner, we had an interesting conundrum this year in that our judges actually were unable to separate the two top presenters. And so rather than having a second place and first place winner, we're going to have co-first place winners, and they'll each receive $1,000 for their efforts and for their success in this competition. And those two winners are KT Christensen and Shohan. And at this point, we'd like to allow each one of them, and we'll start with you, Katie, to give a brief set of words that you want to give relative to the competition and those you want to thank. So the floor is yours, Katie.
Well, I'm gobsmacked. To the organizers and judges, thank you for recognizing the often invisible research performed by fiction writers. Thanks to my advisor, Jill McCorkle, and my committee, Bell Boggs and Zachary Beer. Thanks to the rest of the MFA faculty and staff and my cohort for tirelessly offering me feedback on my writing, and for Rebecca Walsh and James Mulholland for your advice and support, and much love to my DNA Surprises community for sharing your stories. And finally, thank you to my beautiful and complex family. This project would not have existed without you. Thank you.
Thank you so much, Katie. And now we'll turn it over to Shohan Nuzaman for his words.
Thank you, Dr. Harris. Thank you, everyone. Thank you, the coaches. Thank you, the 3-Minute Thesis organizing committee. And I would really like to thank my family, my friends, especially my wife, and my supervisor, Dr. Rohan Shiverkar, and also my entire Department of Industrial Engineering. I'm super excited for winning this competition, and I learned a lot throughout this entire competition. Thank you.
Thank you for that. And so I'd just like to congratulate the People's Choice winner, as well as our shared first place winners this year. And let's give them one more final round of virtual applause. And once again, thanks to everyone who's made this possible. And thanks to all of you from around the world who are watching this event and supporting our students as they move forward in their research and ultimately careers beyond NC State. So thank you all for presenting today. And we look forward to seeing you all again next year when we do the 3MT again.